Thanks
Okay,
for
just a
coming,
second.
guys. Go for it, Ron.
Yeah, just a second, everyone. Just getting myself organized here. Okay, so share. So what we thought it might be to kick this off. I just want to make a couple of very general comments. Can everyone see that now? Yeah,
Perfect.
great. So welcome everyone. Please feel free to interrupt and to take part in any way the spirit moves you. And I thought just to get things rolling, as our sort of general topic really is morphogenesis and morphogenesis in the forebrain, I thought it might be at the minimum amusing, but perhaps even instructive to make a few sort of general comments about the sort of big ideas that sort of have at least historically um, influenced the way we think about this question. And for me, and I think for many others, one of the really sort of founding experiments in developmental neuroscience is the organizer experiment of Spearman and Mangold, where they did this in Triturus in amphibian, but they transplanted a small part of the developing embryo, and they found that they could get a completely ectopic axis, which particularly included neural fates, highly organized neural fates. This is extremely influential, this idea, and led to Spearman getting the Nobel Prize in 1935. And up on the top here, I put a list of people, Spearman and his dates, and Mangold. Mangold died very young, which has led to a sort of, I think, a completely unnecessary kind of, well, perhaps not unnecessary, but a sort of exaggerated confusion in literature about her getting enough credit for her work. But in addition to Hilda Mangold, Spearman had other very prominent students. Peter Newcoop and Johannes Saltfretter among them. And I just put their names up here with their dates to give us a sense of time and also to encourage anybody who is interested to look them up in Wikipedia. They're extremely interesting figures, and they've independently made really fundamental contributions to our understanding. So as I said, Spearman, a really interesting kind of complex figure historically, but introduced this idea of an organizer acting on other cells to pattern them, which was the key feature of why he won the Nobel Prize here. But in the year after he was awarded the Nobel Prize, Waddington, whose name is also very well known to developmental biologists, has a paper in Nature on organizers in development. Now, it's very short. I think it's worth looking at. It's about the role of organizers in different species. Do organizers exist in other species aside from amphibia? And this whole topic is discussed in a more contemporary way and really quite a detailed academic way in this review from Alfonso Martinez Arias and Ben Stevenson, which I also recommend to you. This is quite. Yeah, I used the word academic before. It's a very well written uh, academic discussion of what organizers are. So, in addition to the notion of organizers specifying the patterning of the forebrain, another very important idea in our understanding of developmental patterning is the notion of reaction diffusion mechanisms, which was introduced into biology, interestingly enough, by Alan Turing. And you can see why Turing was the originator of this idea when you look at this simple model here, which is basically a logical switch. But Turing understood that such switches can create patterns in space. And his influential paper was titled "The Chemical Basis of Morphogenesis," published in 1952 in, I don't know, the Journal of Theoretical Biology or something like that. Um, an extension of this general sort of idea was introduced by Lewis Wolpert, who was a developmental biologist, South African originally, worked in London at University College, who was very interested in positional information and was very influential, and used the notion of a French flag, which I've sort of written down here just to remind us, and next to a representation of the French flag, which comes from a Peter Zanstra paper, which is much more recent, which is using the notion of reaction diffusion and positional information to talk about the organization of the very earliest stages in pluripotent cell differentiation, which is just represented here in this model, where he hypothesizes and experimentally tests that notion that these interactions between BMP4 and noggin lead to the positional patterning of cells, so that you get CDX2 positive, very, very early cells, brachyury cells somewhat later. This is the T locus, the famous brachyury locus, and a SOP2 domain. Many of us, I'm sorry for this movement, many of us who work on very early pluripotent cells will find this extremely interesting and stimulating. So I want to go now from these sort of very basic ideas to show you what happens when you apply these kind of ideas to the gastroloid system. So we're interested in, at least I'm interested in the forebrain predominantly, but many, many people now are working on the differentiation of posterior face, which I've used the word gastroloid as a general term to describe them. And a relatively recent paper shows that if you make a group of unpatterned cells, which they call an embryoid body here, and you put next to them a group of cells which are signaling with BMP, and opposite that, a group of cells that are signaling through sonic hedgehog, you actually pattern the undifferentiated cells in the embryoid body in a way that gives you all the cell types of the neural tube, assessed by single cell sequencing and by immunohistochemistry. Really quite a remarkable thing, because actually, as far as one can tell, at first order, it's a complete patterning of the cell types of the neural tube. Just with two organizers, these embryoid body cells are not actually pluripotent at this stage. They've driven them into a posterior fate with a number of signals, including retinoic acid. So over here, I've just shown you a number of still images. And if you look up this X file here, you can find a video from this group, which shows the way the somites are extended through time. So here we have essentially a pseudo two-dimensional array of fates across the posterior dimension of a vertebrate. I'm going to say all vertebrates are going to share this, or many features of this common system. So here's an example of a very early representation of the skeleton of a vertebrate. This is one of the first accurate drawings of the skeleton of a fish done by an Italian graphic artist in around 1600. And I just put up here the plaque on the Boulevard Raspail. This Raspail was one of the first people who understood that all organisms are made of cells. So 150 years before that, people are drawing skeleton of the fish. And so this anterior posterior organization, which is so beautifully illustrated here, is what I wanted really to use these images to emphasize to you, that gastroloids are generating these posterior domains. Exactly where the border is between the gastroloid and the more anterior headlight structures, I don't really want to get into. But if you're really into it, I'd be quite happy to talk a little bit about it. But you can see the point here that even though these posterior domains are highly organized and conserved, they're not completely conserved. And even with the expression of a morphogen like sonic hedgehog, there's clear evidence that different species of vertebrate use sonic hedgehog differently in the specification of this posterior domain. And it's this variability, I think, that's really the most interesting feature of this system that we need to explore next. And I sort of represented that in the title to this image, where I say we want to comprehensively model patterning diversity in the anterior neural tube. How different are we from each other, actually? Now, we've made a little bit of early progress on this kind of question. We've looked at the expression of different patterning genes in early pluripotent cells. This work was discussed in if you go to the Carlos website, you can find a seminar on October 25th last year, where Sulki Kim talked about this. And I just used these two images here to represent the 100 human pluripotent cells. This is the expression pattern of SOX21, a forebrain specifier, and the retinoic acid receptor B, a posteriorizing signal, are expressed quite distinctly in this population of human pluripotent cells. And these two patterning genes, very quickly through gastrulation, this is gastrulation in the mouse, are represented either in the forebrain or the hindbrain. So this is clearly encouraging us to believe that these organizer type signals or morphogenic signals are acting in some way very early to specify fate. But they're not only acting early, they're acting at later stages than in the development of the forebrain in this case, to specify either dorsal or ventral neuronal fates. And that topic is discussed by Nicola and colleagues in a paper published in 2020, where I think one of the key features of this paper was to show that actually when you grow pluripotent cells, they specifically move through organizer states before they generate neurons. And cell lines that are going to generate predominantly excitatory neurons or predominantly ventral fates can be distinguished at very early times in this process by the expression of patterning signatures. And in more recent work from Nicola, which we'll talk about a little bit more detail, Nicola's gone and looked in the developing macaque telencephalon, and he's done single cell sequencing of a number of different domains in the developing forebrain of the macaque. And he's defined that way different signatures which represent patterning cell types, organizer cell types. And here in this UMAP, you can see the distinct representation of HEM cells, anti-HEM, zona limitans, intrathalamica cells, a number of different cells from either the anterior or ventral ganglionic eminence type structures. These are grouped together, and they're actually functionally related to each other, at least to some extent. And I want to close this sort of general introduction by saying that these signatures are expressed predominantly very early. So this is a representation, a bar diagram of the intensity of the signature. Sorry, and early times. There, if you see here, this. I'm just trying to move this pointer very slowly so I don't move the slide. Embryonic day 37, you can see the signatures are present. At 42 and 43, they're significantly diminished. And by the 54, they've essentially absent. So these are transient signatures. And I think I'll just stop there. The purpose of this really is just to give us a general kind of sense of where we're going to, where to start together, thinking more about what morphogenesis means when we're thinking about the development of the forebrain. And And let me just say again, I'm really excited that a number of us are getting together in this way because there's so much data now, and it's really impossible, in my humble opinion, for any one of us alone to handle this data. And right now, there isn't an artificial intelligence algorithm that can do it for us. So we, we're relying on each other to create the right kind of structures and thoughts, which will give us real insight into how to comprehensively model for brain development. Okay, hope you find that useful. As I say, I'm really looking forward to this discussion today. Thanks so much.
Thank you, Ron. So with that, getting our heads going on what morphogens are, how cells organize using them, and variation not only across species but across individuals, I would like to start guiding us through some of the papers that we've had covered in the series. And please interrupt and jump in as much as possible. I'd like to talk as little as possible while maybe guiding our conversation. So I'll start a little, and as I start to show things, please do jump in. I'll be using Nemo now to kind of bring dynamic data exploration to the notions Ron has brought us up with, and then also as we go through the individual papers that many of you have published. So I will go ahead and start sharing now. And so this is one of the studies Ron alluded to, some of our work, Silke Kim leading it, on the very earliest steps of lineage emergence and differential lineage emergence in different cell lines and different individuals. And these are some pictures of from the paper that I kind of want to use to bring us into the data environment with a sort of a biologically driven trajectory. So in the early stem cell model that we were using, it was a monoculture model looking at pluripotency and morphogen treatment, either dual SMAD inhibition via NSB or BMP treatment. We had bulk RNA sequencing and decomposed it into modules and pieces. And we could look at those modules in the data itself or in their parallels in vivo data. And this in vivo map that I'm pointing to here is from Bertie Gutgen's study in 2019 of single cell study of the mouse embryo, where we can see the dynamics that are emerging in the in vitro human pluripotent stem cell system as lineages are committed to. And we can see them flow through the emerging mouse embryo. So jumping now from the paper right into Nemo, and again, jump in as we go through these. This is the data from the paper, and I will share all the links from today with everyone. Everything I show is public and live online and explorable to anybody. So this is just showing the expression of GBX2, which many of you are familiar with. It's involved in the midbrain hindbrain border at the OTX2 GBX2 sort of edge that is formed, and different lines that had different propensities for anterior or posterior fate emergence. Even in pluripotency before we applied any dual SMAD or morphogens, the lines are here colored differently. In cycling within pluripotency, this blue line, I4, is the line that ended up being the most posterior biased. And already within pluripotency, we could see this, basically why it's ready to do this. It's already idling the expression of GBX2 at a higher level than all the other lines. And the most anterior line here in red, SAO1, has very much lower OTX2 expression. Again, in pluripotency before any signals are even delivered to these cells. So this is just to show you this is explorable. You could type in any gene name here and look at it. The other data sets here, each in a different frame, are from that study, as well as these that Ron showed from, this is Ihor Limishka's data set that Ron mentioned has 100 individuals, 300 lines from those 100 individuals. And in their data, you can also see here where in this box here, where each dot is a line, any dots on a vertical line are lines from the same human, and they're arranged in order of their first principal component. But you can see GBX2 expression has sort of an active portion at two of these places, and the coloring here is by retinoic acid gene expression. So GBX2 is expressed at higher levels in iPSC lines that have high idling retinoic acid signaling. And GBX2 is a well-known target of that retinoic acid, posteriorizing, famous for being a posteriorizing morphogen. So again, here, just bringing together data resources to look at very simple things, the expression of a single gene. But the power here is that we know the developmental biology of GBX2 and of these morphogen gradients, so we can impose that biological concept as we look through these data sets. And maybe lastly, I can bring us to the mouse embryo data from Pijuan Sala, where we can see GBX2 expression in the emerging embryo in different portions that will become the CNS, and marking those posterior domains. Okay.
Carlo, can I ask you, like, when
Absolutely.
you show the average expression of retinoic acid genes, it would be possible to plot others, like, for example, Wnt genes or...
Yeah, so absolutely. So in NEMO, in fact, that's one other thing I can do. I'll go to the next page right here. This is the exact same collection of data sets we have, but now instead of looking at a single gene, I've uploaded the dimensions that we broke that bulk RNA-seq data set from the Silkes paper into. So you can upload, whether it's your list of favorite Wnt signaling genes or any other list of genes, and that can be as simple as a list of gene names, or it could be complicated as a genome-wide list of all genes, each with a weight. And then you can visualize that signature in all these data sets. So here we're looking at pattern 12, which back here in Silkes' paper is the sort of slow arriving neurectoderm pattern that shows up exactly in the presumptive brain of the mouse data set. And so this is the static figure from the publication. And we've now have that data and those signatures here. So pattern 12 in Silkes data here, you can see lifting off to variable extents in the different cell lines under dual SMAD inhibition. And as we go through these other data sets, then I'll come down to the ones we know. Again, here, in these two areas where retinoic acid gene expression was high, one of them is related to this neurectoderm pattern 12. The other is actually not elevated there. It's actually elevated more in more mesodermal, even more posterior fates. So for sure, you can, then that's exactly how we want to, as a group, start to think about this. With data sets of interest that we get into NEMO, what would you like to see in them? And we'll walk through more of these right now as we walk through some of the papers. And in fact,
Arlo, can I
yes.
just contribute? So, Soraya, you can also very easily just search for individual genes, and you'll spend a lot of your life being absolutely absorbed by these patterns. It's extremely interesting and easy to do.
Yeah. And actually, Soraya, I remember just the other day, in fact, maybe we can jump to your paper to start this going. And maybe do you want to give us a 3-sentence introduction to the morphogen chamber, and maybe so that we could introduce the idea of your modules here and what these patterns might mean if we're going to look at those in other data sets?
Oh, sure. So like in the lab, we developed this passive diffusion device that allow the formation of gradients along two major axes, dorsal, ventral, and anterior to posterior, like as you can see. So in the central area, we basically embed the embryon body, so the pluripotent stage, and just 5 days of exposure to these orthogonal gradients, we were able to pattern different brain regions And this was the patterning acquired early on was quite stable also at later stage. So what we wanted to know, like also the device allow us to analyze discrete position within the culture area. If you can see, like there are 5 consecutive chambers, like this C1, 2, 3, 4. If you can see my cursor, like in this way. And so this could be further subdivided in dorsal and ventral. So we have 10 positions. And so what we did by bulk RNA sequencing, Alexandra actually dissected the expression profiles that could then be related to later stage cell types that we could identify by bulk RNA sequencing. So for example, if we focus on the pattern 1, 2, and 3, like you can see that involved genes that were like highly expressed on the most anterior part of the device, and actually they were negatively regulated by Wnt, because more we go close to the source of Wnt that is on this side of the chamber. Here there is basically high Wnt, imagine, in this way. Basically, these genes were downregulated. So similarly, instead on this side, if we focus on the pattern 17, 18, and 19, actually this group of genes were actively stimulated by the sonic hedgehog. So of course, it's not easy like or black and white that we could quickly classify like only activated by Wnt. There are other many other combinations. But we found very interesting that these patterns were predictive of the later on cell types that each line was able to generate. So if you go in panel F, ENF, we use this pattern to characterize the line, similarly how you did with in Kim papers, that we identify lines that were more prone to posteriorize. For example, in this case, the line in orange was already at different level for those genes that were activated by the Wnt pathway. So it was on average, also we quantify like the percentage of cell types of hindbrain here on the pie chart in pink. So this line was generating more posterior lineages compared to the others. And similarly, like we found other lines that instead were more responsive to sonic hedgehog. So basically we identify a line-to-line predisposition or readiness to response to morphogen.
Very cool. So then to bring us sort of to the live version of this is now a collection of data sets, including your own, Soraya. We'll get to it in just a second. That in NEMO, just to explain to everyone, in general, you say what you want to see and then the data sets you want to see it in. So this can be a single gene or a signature, as we were talking about. And this is a collection of data sets. And here the ones I've brought together are relevant to regionalization. This is Linnerson's mouse data, Nicola Micali's data with Sestan and Pashko Rakesh here in the developing macaque telencephalon. And this is again from Linnerson, the more recent Brown data from early human development. And then below this, we're getting into some of the studies that we've had in this series. This is the Neil Ammon and Sergio Pasca data set. They have 2 data sets. This is the data set that Soraya was just talking about. Her day 9 data here in bulk, where each one of these units is the chamber she was referring to, with the sonic hedgehog source down here and the Kier Wnt source here. So this is dorsal and this is ventral. And then the Wnt gradient goes in this direction. So this is showing the expression of TTR, which is a choroid marker and a significant organizer in the early brain. And you can see not only is it, and this was actually surprising to me, it's quite heavily driven in the ventral domain here, I would have actually guessed at the opposite. It's often associated with the dorsal hem signature. So we can talk about why and how that might be happening. And you can see here, many days later at day 64, the TTR and choroid identity in a very sort of specific pocket of these day 64 cells. Again, just to show you kind of how these data work together. And coming back to the Neil Ammon study, they don't use a gradient system. They used many different combinations of morphogens. And here's the single cell data and a UMAP, and that same data simply in a violin plot. So we can see that in a very discrete section of the cell map, there's a high TTR expression with a little cells that they call midbrain here. So, and again, TTR isn't anterior or posterior, right? Anywhere where a ventricle is and there's going to be some choroid, you'll see it appear. But here, when we break it apart by violin plots, it's easier to see what treatments induce it. And TTR is quite specific here. A couple of the different BMP4 treatments induce that gene, but not always. If you combine it with sonic, the SAG is a sonic agonist. I think it's a smoothened agonist, right? You don't get TTR induction. So it's these instructions are not simple, and in time and combination and in space, all of this is dynamic. And again, this might be very surprising given that in Soraya's data right here, it looks like sonic signaling is turning it on, but this is quite early. So that might be a different spot, because here,
Yeah.
the SAG is not inducing it, at least not with BMP, and none of the other SAG conditions really kick it up. It's these KIR plus BMP4 that's kicking it up. So it's very interesting to see parallels, but often with quite subtle differences in these models, you really see surprising things. And again, time, space, and line is all introducing quite complex variation here. And bringing all these studies together to look at them this way, hopefully we can start to pick
Can
it
I just
apart.
make a comment about this, Carlo? I thought about it a little bit.
Yeah.
So the data from Sergio's lab, this data we're looking at here, Amin et al.
Yep.
It's done quite late. I mean, I can't remember the exact days, like 64 days, or it's not at early
Is it 64?
times.
This is the paper. Yes, that's right. That's a big difference between the
right. Whereas the data that you were showing from Soraya's work is on day 9.
Yes, right. Basically, almost right after patterning.
right.
Right.
And then here
So
we
I
have
think,
the later one. Yeah.
So I think what's very interesting is that TTR is a component of the choroid plexus signature, and it looks like that choroid plexus signature is extremely stable. So even when you go out to late times, you can see not only TTR, but you can see. So that's that where you put the green arrow. Yeah, that's at late times.
Yeah.
But you can see the whole signature in the choroid. It's as if the choroid is turned on by BMP, and then that BMP-driven signature stays on. But here up above, it looks like there's some kind of, at early times, there's some kind of a sonic hedgehog type involvement, which is, I think, the real challenge we have. It's extremely interesting. Okay, I hope that's helpful.
Yeah. And then go for it. Yeah.
I just want to comment that also in our case, not of course consistently, but also KIR has a kind of an influence, because this is also the position close to KIR. So both.
So high Wnt is also doing it?
Yeah, not always, but I think this pink means it's higher, right? Like,
yeah, the light pink, that's exactly right. Right,
yeah.
right here.
So it's not always, but in some case. So
yeah, okay. And then there's one line that seems not to care much at all.
Yes, this is the line that want to generate cortex at all costs. Yes.
Very cool. So to extend the exact sort of notion we're thinking about here, this is TTR, a single gene. Here are the same data sets, But these are Soraya's 20 morphogen patterns from the paper right here that we were looking at. So these 1 to 20 patterns, I loaded up into Nemo from her supplemental tables. And so now these are now visualized on that same data set. So P2 was one that in her paper you just mentioned to us, Soraya, is one of these up here at the top.
Enriching cortex, yeah.
Yeah.
That probably determining like cortical fate. And it show also good correspondence with the brown atlas, yeah.
Yeah, so here it looks like high in cortex and hippocampus, especially the pattern 2. And so if we come back to the live version on Nemo, this is literally what is displayed here on average, right, as the average of all, well, actually it's pattern 2, not 1, right? Let me get that right. This pattern, which is extremely high left, which is the lowest Wnt concentration, right? Here, that is basically the average of all of these maps, correct? It should be the same thing, right? So that's why we see here at the left, it's always on. It's always on. It's always on. It's always on. Here, it's always on at the left, but maybe with a little bit of variation here. But if you averaged across all of these, the thing you really see is this in the lowest Wnt positions is where pattern 2 is really, really emerging in the early data. And then we see.
Keep in mind that sonic hedgehog is not added to the first compartment on the left to V5.
Yes. So that is a combination of the lowest Wnt as well as low sonic, even though it is ventral.
Exactly.
Correct? Yes, that's a point that, Soraya, you made very clear to me the other day, reminding me of this initial picture here, where this is actually a domed sonic gradient. So this last position is not necessarily high, it's low sonic, right?
Yes.
Yeah, that's super important to understand this map, right? As we look at this, this position is exactly what we're talking about. Although it's the same here usually, so it looks like it might primarily be the Wnt in this case and not the sonic that's driving it. But
so the reason why we did that, it was because this notochord, right, which is the origin of sonic hedgehog at the very, very beginning, is actually barely extending. I mean, Ron can correct me if I'm wrong, but I think it doesn't extend beyond
something like this, right?
The diencephalon, yes.
Right.
On the other bench.
Yeah.
That's exactly right. That's actually extremely interesting. So in regions that are posterior to the diencephalon, the floor plate is a very narrow, well-defined group of cells that are making sonic. In the diencephalon, though, that domain is expanded, and so in the spinal cord, the floor plate doesn't make neurons. In the midbrain, the floor plate makes dopamine neurons. In the diencephalon, the floor plate makes a lot of neurons that have absolutely crucial roles in the brain. So it's really worth emphasizing the point Flora was just making, that the notochord and the floor plate being a kind of CNS avatar of the notochord is a very interesting domain. Yeah.
Well, and in fact, in that region develops the basal ganglia, which is a forebrain structure. And so I believe there is a little bit of sonic hedgehog there too by diffusion, right? But there is not an autochord, so perhaps the concentration is a little.
So in the frontal region, where the GABAergic neurons, there the sonic hedgehog domain is extremely precisely localized. That the person to look at there is John Rubinstein's work, and it's positionally very precise, and its expression of sonic hedgehog is well defined, at least in the mouse.
And to add some temporal thinking to this, in some of our work, I think it's the Mikali 2020, sonic is not the first thing to come along when you see the ventral signature, which I think if we're thinking notochord and spinal cord, we often think of sonic as the initiator of the ventral fates. That game might change up front more anteriorly as we're talking about. And we've actually seen transcriptional programs of the ventral identities coming up before sonic is expressed and sonic following that. Now that is in vitro, so we might want to see if we can see an in vivo correlate of the timing there. But just to bring up the notion that the dorsal ventral game or the floor plate notochord sonic signaling game is fundamentally different in the spinal cord and the telencephalon, as you guys have really been focusing on.
Well, I think just to continue this, guys, it's up to us to show what we mean by fundamentally different. And actually, in Nicola, if you're tuning in, one thing I should be able to figure out from your paper, but I couldn't get it at first glance, is that it's not every patterning center in that ganglionic eminence grouping that's actually high in sonic hedgehog.
No, right. You're right. I mean, sonic hedgehog. We had we annotated the ganglionic eminence cells as a patterning center using the NCase 2.1 marker. So if you Carlo, maybe if you share if you open the link here, if you click on the link for the website, you can see that as in vitro, sonic hedgehog in our dataset is expressed in later than in neurons, in interneurons. So I mean, again, at this stage of the monkey, okay. So if you go earlier, probably you can see that sonic hedgehog is expressed by more stem cells. So no.
Oh, that's interesting.
So Carlo, please. No, the website now, you can also use this one, but if you share the website from the science paper, please.
Oh, the one in your...
Right. So we scroll up.
Is the link right in the front?
Right, the link, right, exactly, here. It's page 2, right.
Page 2. This one?
Here.
Oh, you got it.
Click here. Okay, then here. Okay, so now if you do also the circular, you put here sonic hedgehog. Plot. You see? Sonic hedgehog is, it's very early, but it's expressed by neurons. Here, it's very early, as you see here, E37 and 42, is ganglionic eminence here in purple, but it's expressed by neurons. It's not expressed by the stem cells. And so basically, these radial glia cells that we annotated as pattern centers, ventral pattern center, ganglionic eminence, as ganglionic eminence, those progenitors that we basically consider pattern centers, were basically annotated as NCase, where basically we consider the NCase 2.1 positive progenitors. Not sonic hedgehog positive, because we
aren't these relevant to you?
Sorry?
But isn't KX2.1 just downstream of sonic hedgehog, according to
Rubinstein? Yeah, but actually there are also other papers showing the opposite. That NCase 2.1
so what
sonic hedgehog.
So Nicola, what are these early neurons expressing sonic hedgehog in the basal ganglia? What type of neurons are
Carlo, can you scroll up? These are we annotated these neurons as interneurons here. What is it here?
Yeah, LHX6. Wait, well, aren't these interesting too, though? These are the early subtypes these are neural stem cells that are coming on before this.
Can you scroll down?
Yeah.
Yeah, yes.
But
Can I
that's
make a
But
comment
they are
about?
they are low, low number of cells. These early subtypes are very low number. Yeah.
But that's what we would expect from any guidepost organizer, right?
Yes.
But I want to just interrupt a little bit. So,
But anyway, you see that the key is higher, the expression is higher, no?
Much higher, yeah.
but I think it's really important to understand that we're looking at a dynamic system, and the system is non-linearly dynamic. And if you're interested in Sonic Hedgehog as an example of this kind of nonlinear dynamics, I highly recommend reading John Rubinstein's paper on NKX 2.1 and Sonic Hedgehog. It actually flips. And so that's kind of why I think it's so valuable to talk like this. Because when one's alone thinking about it, each of us tend to get stuck in certain kinds of views because we have to simplify the problem in order to grasp it. But I think this discussion we're having now around Sonic Hedgehog is just a beautiful example about how valuable it is to really think together about how these systems actually work and what we really need to do to figure out how morphogenesis is actually controlled.
Carlo, I also want to tell you that I'm talking about, here I was talking about ganglionic camelids. These cells are not from the ganglionic camelids. In fact, these cells are annotated as anteroventral domain, organizers, but anteroventral.
Yeah.
In fact, you see that, I mean, at least it is a true anteroventral from frontal regions in red. I mean, in particular,
So Nicola,
this one, yeah.
of course, you did all the dissection here. So we're listening to you carefully. But if you go and look in the mouse, Sonic Hedgehog is not expressed in the ganglionic eminence.
Right.
It's expressed in a region which is just adjacent.
Exactly.
In the anteroventral area.
So in that case, what you're saying is this is the same as in the mouse.
Yeah, exactly. I mean, this again.
Well, that's perfect.
We did not find Sonic Hedgehog in the stem cells of the ganglionic eminence.
That's correct, because Sonic Hedgehog is the patterning gene.
Right. They determine, they specify the ganglionic eminence. They are not the ganglionic eminence.
Right. Okay, thank you very much.
But yes, but the ganglionic eminence here, we because we are talking about very early time point. We basically, our point was to basically we included these cells are pattern centers because they are very early and they are ventral.
Yeah.
Just one thing that needs to be kept in perspective is also what Kiker and Lumsden in their 2012 review talks very nicely, the effect of Wnt inhibition from anterior neural ridge and the cells for Sonic, the cells become susceptible to Sonic way later as compared to diencephalon midbrain when the initial patterning is observed.
Time and space.
Yeah, and Wnt inhibition.
Cool.
So to take a peek at this, and now I've loaded the Sonic gene into these data sets, we can see where it's showing up early on in the mouse data, where these are in general ventral fates and these are dorsal fates. And that's not where it's expressed, right? It's expressing it before those fates to direct things. And then here, let's also, let's go further down. Something that might be interesting that we haven't, if we keep coming down, well, actually, this is interesting. Sonic is in a feed forward positive loop with itself. When you stimulate Sonic, you get Sonic in the chamber. Down here, I've also have some incredible spatial data. This is StereoSeq from Chen et al. in 2022, where we can really see the tissue organization of some of these. Sonic itself looks like it's a little low. I should have kept TTR up here, actually. But what we can see, let me jump now to Soraya's, I think, where did I have it? Is it all the way up here? Oh, actually, let's do this. That's here. So we've also loaded those 9 patterning centers from Nicola's monkey data into NEMO so we can visualize them. So this is the monkey data from where it came. So these two stripes are the same stripes you can see in his paper called Patterning Centers. And this was from that subset of these early states looking specifically at the different patterning centers. And so each one of these clusters were used to generate these gene lists. So this is kind of a circular thing just showing where the genes came from. And if we come down here to the in vivo spatial data, of course, we could look at any of these data sets this way. But early on, it's a little bit hard to see. It's not as clear here and here. And this RSPO3 is the hem signature from the telencephalon in the macaque. But as you go forward, now we're into day 11 and a half and 12 and a half. This dorsal, very restricted hem signature is quite crisp and clear, even more so in these sections that have the brain elements dissected out. And then it just like most organizers, once you see it very clearly, then it disappears. Once you get the sort of mature structure of things and everybody knows where they are, those organizers tend to disappear. So again, this is just to see this, but also to show us how, if we have a set of signatures or modules we're interested in and relevant data sets, how we can combine these to do some exploration. And now, if we jump back into Soraya's system that we've been looking at here, the hem signature, not at all surprising, is much more highly represented in the dorsal, where the sonic signaling is low. It also looks like low Wnt helps a little bit, although this one is backwards. So there's a good bit of, even though there's a clear dorsal patterning or dorsal bias here, the different lines have different ways of getting there, or different patterns.
What are you looking at here? What's the gene that you're looking at?
So this is Nicola's hem signature from his monkey data.
Oh, so it's a number of genes.
Yes. And in fact, on this page, you can always come right here to the top, and it'll list the top couple. But if you click on this button, this is just, there's no fancy weights. It's just 0 or 1. So every gene with a 1 next to it is in this group. So it's quite a big pattern. Looks like somewhere near 100 genes. But here are those genes, and you could download them or just simply look at them here. But that is the packet of genes that we're now looking at in Soraya's data, captured from the monkey telencephalic single cell data. And very commonly, this is something we see. If you remember, the TTR and choroid signature was right here, and the hem, which physically is right next to it as well, is also very close to it in its molecular signature, and therefore in the day 64 data from Soraya's paper is right next to that choroid signature.
May I ask a question to Soraya, Flora, and maybe Koraf, since you all work on microfluidics, and now that we're talking about the temporal aspect, did you ever try gradients over time? Because in vivo, we're also like used to, like we're simplifying the system and trying to think of like concentration steps, but like those concentrations vary in time, and like a certain position, like a certain cell is exposed to like varying concentrations over time. So did you ever try to model that, like increasing, for example, the amount of sonic that the organoid gets?
We didn't, but basically we started with the highest concentration. But of course, it's possible. So the advantage of the system is tunable, because each reservoir that's around the chamber allow us to, we could add even more morphogen. The importance, of course, is the molecular weight. To guarantee that they will form a gradient, because if it's too small, will diffuse fast. So we can tune though, because we could change the media more frequent.
Do you remember when you increase the, what was it, sonic hedgehog, that you moved the hindbrain?
Yeah, the wind,
was
the
Kier.
Kier.
Oh, the Kier, yeah. So moving the wind, move the boundary.
The boundaries.
OTX2 and GBX2 rostrally, increasing Kier concentration.
I think more a question is if in times we play with the morphogen, but we didn't do it.
No. No,
that
would be worth doing it, but
basically we try different paradigm like 5 days, 7 days, 10 days, and what we were amazed that just 5 days was able to achieve all those diversity. So we thought that could be like advantage that we don't need to, because of course all those morphogen are costly and if the cell seems, but of course for a fine, if we want to study kind of these temporal dynamics, we should try to change it. Yeah, because of course, like while the neural tube elongate, maybe the cells becomes like further from the source. So many things could be adjusted.
Because yeah, we tried a little bit to look into this, but our system is not the best. Like with organoids and with like static concentrations for each of them, we don't have a way to like gradually increase the concentrations, but rather like do media changes and like go like one step above. So that's why I was curious if you tried or Gaurav, did you ever do this in the Mister?
I mean, we did it together with the telencephalic Mister.
But over time, like increasing concentration over time.
No, not really, because I mean, I think the idea of Sonic was very much based on the Parkinson's protocol, and we were playing more around with how much of Sonic is good enough so that it kind of gets. So Sonic is a hydrophobic compound, and PDMS absorbs it a lot. So without losing a lot of Sonic, how best we can pattern and get the ventral floor plate for the RC misters, but for the telencephalic mister, yes, we had to, not increasingly, but we had to try multiple combination and multiple timings to get the Sonic right.
Yeah, I mean, this is like a whole thing by itself. I think like it's like step by step. Like once you have everything like narrowed down to concentrations that work, then you can play with this. But yeah, thank you. I was just curious.
So one other.
Are you still there?
Go for it.
Maybe this is a good time to show that summary of all the patterns, Nicola's patterns in the,
great.
because I just to reinforce the point that Carlo was just making. Here's an image which is just very similar to the one you were looking at.
Want to hit that share button, Ron?
Yeah.
Great.
What you were just looking at was what was the TTR signature, right? This one here. I can't remember. What were we looking at?
The RSPO3, the HEM. Which was
R-spondin-3. Yeah, there we go. So, yeah, we were looking at this, and Carlo was making the point that this is in the dorsal signature. But what is very striking when you pull out all of these images from of Nicola's patterning signatures in Soraya's data, you'll notice that all of these ones have a really strong sonic hedgehog component to them. It's really beautiful. I just thought it would just be an appropriate place to show this. It's not a one-off thing.
No.
That it's clear that the signature is being induced. At least I think it's clear. Maybe you can help me understand it. That's my first grasp of it. Again, not all the lines are exactly the same, but overall, it's this lower band here, which is showing the strongest expression. But you were saying, Flora, that that this furthest left
doesn't have a
chamber doesn't have sonic edge at all.
At all, yeah. But actually, because it creates like a gradient that is a dome shape, it will receive a lowest dose from the nearby chamber.
Right.
But yeah, this line, especially this last line, always in that position
is empty.
Recognizes that it responds to that difference in sonic, and that's the only location in the ventral position.
I find this very exciting. I mean, this goes right along the lines we were just discussing about the quality of the data that we're generating here. anyway, you'll notice on the right-hand side that it's a very different pattern, right? That these other patterning signatures, I don't know whether they don't show the same
A lot of line
effects
effects.
as you see on the left-hand side. Right, but there's a lot of variation and there's a lot of interesting things to discuss.
Mhm.
Yeah, a couple of them are more driven by line than they are by morphogen, which is kind of astounding. These two in particular.
I have a question for Soraya. This TCF7L2 at the day 9 data, do you see it specifically for ZLI, or you see it like in the entire gradient?
Basically, as you can see, so in this case is the pattern that comes from Nicola, the zonal impedance intrathalamica gene list, and is interrogated in hours. So it seems and kind of correspond because the line 1120 is the one that tends to posteriorize. So it seems that this early stage at any position is having this high expression of this patterning center.
It's very hard to see the name of the lines.
Yeah, is it this one that you're talking about
It's
here?
the third one. Yeah. The third one. Is it you, Carlo, that is sharing?
This
Or
is
maybe,
Ron's
yeah, we can
screen.
see. Oh, yeah. Perfect. Yeah,
Can you
so
see
what
them
we
now?
know, yeah, what we notice is like the effect of sonic hedgehog. We don't know, probably it could be also the concentration that we chose. Make like the line less variables. So it seems they are more responding similar to the same. And instead along the other axis, like when the axis that is governed by wind, they show more variability.
Yeah. You're making a really interesting point now, I think, because as Carlo was mentioning a minute ago, Sonic Hedgehog is turning on Sonic Hedgehog.
Mm-hmm.
So if you're looking at something that's turned on by the ligand, then all the lines may show a more homogeneity. Whereas when you're looking over here, right,
Mm-hmm.
now you're seeing the sort of intrinsic variability in the line in the absence of a homogenizing force, morphogenetic force, right? Do you see what I'm saying? I think you're making a very interesting point, which is really relevant to us. BMP turns on, BMP4 turns on BMP4. That's part of Zanstra's logical gate. Right? That's how these things work, really. So I just want to emphasize the point you're making is really relevant to us, and we could use it to our advantage.
Yes, I think a lot of the ideas we're talking about can lead us to thinking about assays in two ways, where a lot of efforts have been dedicated to assays that reach homogenized endpoints across many lines. And there's many reasons to do that. So you have a cell population from many different donors to test. But on the flip side of that, and this is something that Ron and Soki and Nicola and I have thought about a lot, is maybe also what we need are a bunch of assays that do the exact opposite, that in any one line have a repeatable result, but have highly variable results across lines. Now, that doesn't give you homogeneous cell populations to do further tests, but if you're looking backwards for the origins of variation in people, that's the assay you want. You want to know the difference between people and why one's making more cells of one kind than another. And the design of those assays would be fundamentally different, right? You want them either at a stable energy minimum where everybody's making the same cell line, or you want to put them at the top of a peak where everyone seems to be falling off in a different direction to show you their inherent biases in these molecular signaling pathways.
I would say also,
yeah.
Sorry,
Carlo. Go for it, please.
Regarding what you're saying now, for me, the question is like, how many variables do you need to measure to actually predict the outcome? I think that's what we would all like to know. We were also trying to, in our revision, we're going along the lines also of Soraya's paper, like trying to tackle variability, interindividual variability and reproducibility. And we also try to like have like snapshots about patterning, how each line patterns and everything, but we don't know where all the variability is coming from.
Yeah.
I don't know if like I used to think that it was all like epigenetic, but like is it epigenetic? Is it genomic variants? Is it like what is the feeling that you all have? Like what do you think is the most important thing to look at? Or like can we actually look at one thing? Do we need to look at all of them? We need to build like extremely large models to predict, or like is there an easy way to simplify all of this? What are your feelings that you got from your experiments?
So
I
in
can just
this,
also quickly
go
follow up
for
with
it,
the same
Gaurav.
question so that you can answer both of them at the same time. As other than this, we have also observed that same line, two different grades, like H9 GMP grade and H9 non-GMP grade, respond differently to different sonic concentration. They don't the they don't ventralize that easily, the GMP ones. So what is exactly the correct way? Would it be always we should go like the way Pollen Lab does, that pool 14, 15 different lines together and just do one diff?
Yeah. So
Can I
these are
just, before we go away from H9, can I just ask you to be a little bit more precise about this? So H9 is really a fascinating cell line. And as far as I understand, H9 is still the line that's used to generate dopamine neurons by the Blue Rock Consortium.
Yeah.
Is that so? Is the GMP grade H9 that you're referring to a specific subline that Blue Rock generated?
I don't
Where
know
did
that.
this come from?
So it's just we ordered, so we were running out of H9 stocks and then we ordered a GMP grade line and it
From
behaved very different.
from
From
who?
V-Cell.
From
From
where?
who?
From V-Cell, the Wisconsin cell.
Oh, I see.
Listen. Well, you would have to, have you sequenced the line?
No, we
genomic.
just karyotyped it.
Oh,
Oh, this is
yeah,
absolutely great. This is a great conversation.
you've got to sequence it because although unlikely that, you know, it
Yeah,
would affect all the
CNVs.
cells, you may have a large event that might affect, you know, we've seen those things. But one important question I like to ask is another one. Have you subcloned this line? Because if you subclone a line, then you're going to have large differences.
Okay. No, we did not subclone. It's
Okay.
just expanded and stored and bought it
Yeah.
out.
Yeah, it's interesting. Because, you know, somatic mutation happen all the time, but they affect a minority of the cells in the population and they're all different. So if you have major effects like this, it would be interesting to explore genomically, see what's happening to that line. And how many passages are you talking about?
I think it's 39, 41, 42-ish, not beyond that.
I see.
Yeah. Yeah, our lab's Parkinson's protocol is based on RC17, the STEM-PD trial with Novo Nordisk. It's not on H9.
Do you have bottlenecks in your protocol in generating this line where the selection processes might occur?
No, not really. I don't do the generating line part. I was mostly doing the bioinformatics and the mixture making. But I mean, and the Parkinson's protocol was way done back when Ignite was postdoc in Marlon Parmar's lab. And it was mostly based on RC. It is based on RC17 only. It's not based on H9, unlike Blue Rock.
And Fatima, to come back to your question of how we, at least in this study, handled what you need to look at, that's why I highlighted this box. In addition to the sort of morphogenetic lineage-related patterns, when we did this decomposition, we got a whole number of patterns that had nothing to do with time or morphogen treatment or lineage emergence, but were simply offsets defining groups of genes that each line had slightly higher expression than all the other lines in all conditions. And so these were evident in pluripotency as well as in differentiation situations. And although we could not check with H9 because it's an embryonic stem cell without a donor human, in the IPS lines we used here, these transcriptional signatures were not only specific to the lines in vitro, but to the parental fibroblasts and to the postmortem brain tissue of these people. So to get at what you need to measure and what you need to look at to make predictions, the transcriptome of pluripotency appears to be a gold mine of understanding individual variation before you even do anything to the cells. But to get to those predictions, you need hardcore developmental biology knowledge so that if you are asked, well, cell line X has twice the GBX2 expression of the other lines, what might that mean? Of course, that's a gene we know a lot about, but the genome as a whole, we really don't know. If this collection of 200 or 300 genes is overexpressed in one cell line, there are hundreds or thousands of cell contexts in which those small offsets in expression and reactivity will influence things. So we kind of have to marry classical developmental biology with high throughput screening of transcriptomes in individuals to combine these. And yeah, that's sort of, and to come to your genetics versus epigenetics question here, we found in this paper alone both of those things where I mentioned it looks genetic if it's in their brain tissue and skin tissue and their pluripotent lines. But then there's two lines from the same person in some cases that behave very differently. Now, coming back to Flora's comment, it didn't look like genetics to us. We did genome sequencing, but we had huge, Sungmei Seo, who might also be on the call today, I'm not sure, did H3K9 histone mark interrogation. And these signatures that look like this of individual people and individual lines really track with H3K9. So it's epigenetically driven. There could be genetics under that, but the answer is that offsets in gene expression controlled either genetically or epigenetically can really be informative. And studying the transcriptome directly, I think, is kind of another level of, we all realized after the human genome was sequenced that we should sequence everybody's genome and understand what all these little variants mean. But that game is actually much more difficult than just asking the cells, what does the grand total of your millions of SNPs, maybe thousands of CNVs you have, what is its functional output in your transcriptome? Which appears to be very accessible to us, all of us, with current present day sequencing technologies.
Hi
Just
guys.
to continue. Oh, go ahead.
Yeah, sorry,
Please.
I just wanted to ask a bit before we shift topics. So I get the point, right? So different lines are responding differently, and we know this, and there might even be this meter that one could apply from control to result, where is the push for each line. But I'm also wondering how important is to think about reproducibility here, because reproducibility then means that every line has to behave in the same way. Why would we even expect that? And I wonder, isn't it important rather to have the sum of all variabilities represented in the data than trying to strive towards that the result should be seen across lines?
Yeah, I think a lot of us might have a response there, so please do jump in. But this comes back to what I was mentioning earlier, is that you have to have a goal in mind, right? You don't, maybe if you want to test everybody's dopamine neurons in reaction to X treatment, you want something that every line gives you the same answer. But if you're designing an assay to tell the difference between humans, you want the exact opposite. Now, you don't want a random answer every time you run a cell line. So any specific line should always give you the same answer, even if a different line will give you a totally different one. Now, that is I think, a harder place even than getting every cell line to do the same thing, because your demands of reproducibility are very high, where every line should have the same answer, but someone with different biases should give you a different one. But I think both, yeah, go for it.
You have to test both things in order to know the answer. You have to test both reproducibility and the differences across lines.
Yeah, and it totally controls what you can, information your assay will be useful for, right?
So how do you want to test it? Do you want to grow them together as a pooled culture, or you can test it independently and call it a day?
Either way, I mean, that's up to you, but
Okay, we got a reviewer's comment that said that, yeah, maybe pool culture would be required.
Yeah,
While we
I
have
think the
previously
answer there
shown.
is we would all, I think, in an ideal world, like to test them individually, but that costs 10, 100 or 1,000 times as much as mixing lines. But then, of course, once you mix, one cell line could easily help another cell line over some hump that it can't get over on its own. So I think we're really just at the beginning of knowing what cellular functions we can assess in those pooled versus solo cultured systems. Because for sure, you'll mask certain disabilities or abilities of individual cell lines if you're in a jungle of other human cells. And I'm surprised you got the comment that you're saying you got. Usually I think it's the opposite, where somebody does a pool and you need to check if they act the same when they're alone. You got the opposite question of,
Carlo,
yes.
Carlo, maybe you can show the experiment we published on the Nature Communication paper, where basically using the Jardon et al. Lines,
Yes.
we basically
Wonderful.
we could actually distinguish different types of different types of donors, right? Because I don't really believe. I mean, I agree with you. With there's a it depends the goal that you have in mind, but I don't think we need to test all the time hundreds of variables, right? It depends what you want to measure. So in our case,
Yeah.
for example, we wanted to understand which line were more dorsal, which line had more dorsal bias versus the lines with more ventral bias. For us, it was actually anteroventral versus dorsoposterior. Let's be
Yeah.
precise.
So this
Because
is the
the
data
axes are developmental axis. We want to understand the behavior of the lines in terms of developmental axis of the brain, right? Of the
absolutely.
brain. So I mean, if you want to see which line have more bias towards the left hemisphere or the right hemisphere, probably is a little bit more complicated. But I'm sure that there are lines with this kind of bias too, right? The point is that what we can measure, what kind of tools we have to measure the bias of the lines. I don't feel, again, now I was
Here, let's show the data. Yeah.
Right. I don't
So
think
this
that
is
we
the
have the
data
tool
set that
to.
Nicola's talking about from Flora's lab, Alex's paper,
Right.
doing organoids in father-son pairs where the son was diagnosed with autism spectrum disorder. And this is the analysis he's talking about, which is in a more recent paper that involves both Nicola and Alex. And I will bring us right to that figure, which is here, where this is a combination of several things we've been talking about. One is the telencephalic organizer signatures from Nicola's paper. That's what's here. And then the data set we just introduced, Jourdan et al. And each one of these identifiers is a family. So on the
SD
x-axis
versus control pairs. I mean, yeah, each line is a family of SD compared to the control, no?
Yes.
Its own control, right?
So the red versus blue is higher in the affected son in red or higher in the father in blue. So each vertical column is a father-son pair, and each row is a gene from a particular organizer. And go ahead, and if you want to, Nicola, you can comment on.
No. Yesterday you described this data perfectly. Please go ahead. So here exactly here you have basically a kind of a dichotomy of the donors, right? You have
Right.
basically half of these donors have of course these have been sorted on purpose to see this difference, right? So this can be actually a variable that this data is showing that at least we are. I mean, I would say that we have the control of this variable. It means that we can at least see which line are more biased towards the dorsal-posterior, to have a dorsal posterior bias versus the anterior ventral posterior bias. Now,
does that, Nicola, does that reproduce some of our finding?
So it is what I was saying. So I'm sure that we could do hundreds of experiments more that we didn't do, but can be a starting point. Flora, I know that we checked if there was several, how to say, if this data could fit with your data. I don't remember exactly what was the result, but I'm sure that we checked if there was some kind of a how to say correlation. Again, we could do more experiment that we didn't do, and it was out of the scope of this paper. But these data show basically that there are different, probably there are different bias in these lines. I mean, which map on the developmental axis of the brain.
Yeah. Also, Nicola, when I saw this, it struck me that it's really an important finding that it's pointing at. That it seems like you've got two general classes.
With these parameters, yes. If we test other variables, we don't know. But for to test when we test the bias of these lines, over the developmental the axis of the brain, yes, we found that there are two types of families. Yeah, two types of donors. Yes.
But is there any other variable that can go along with this?
I don't know. I don't know. That's what I'm saying. Depends on the data that you have. Again, if you want to test if these lines are biased towards the right hemisphere versus the left hemisphere,
Do they differ by head size?
Yeah, the macro normal.
No, they don't. There is no correlation with the macro versus the normocephalic. I think that we checked that very well, actually.
Okay.
We don't know why, but and we didn't continue with this research. But I remember that this is what we checked if there was correlation with macro and normocephalic. There was no correlation.
Yeah, thinking in both directions from this finding, you guys are kind of thinking forward. What does it mean for the physical phenotype of the brain, or even the clinical phenotype? But also we can think in the other direction, the origin. What would be the origin of these outcomes versus the origin of these outcomes? And could we see that in those transcriptional signatures that in pluripotency already that we've seen in other cases? So thinking in both directions would be interesting, because observations like this in vitro are very puzzling right now, right? It's clear that a whole family of kids didn't lack ganglionic eminence or lack a pallium, right? All of these people built a brain with about the right size of things.
But wait a second, this can actually reflect different rate of neurons, right? I mean, do we know if this family have more, glutamatergic and this family have more GABAergic, for example?
I don't think we have imaging in these people, do we?
Right. So exactly.
Right. That would be an interesting correlate. Yeah,
right. It's an interesting. That's why I'm saying that this experiment opens tens of other questions. But and everything we can answer to the question depending on the data set we have. I mean, again, let me reiterate the joke that you can see if if this set of this group of lines. This group of lines has a right hemisphere versus left hemisphere. Of course, it's very difficult. But if you have the data set, maybe you can check. But I don't think we have the data set showing. I don't think anybody profiled the very early right hemisphere versus the very early left hemisphere in the monkey, right? So that's what I'm saying. Depends on the data set that you have. Depends on the data that you can correlate these analysis to.
Could I just extend this conversation just for a minute longer? So what I find myself thinking about is that, as Carlo mentioned, when we look at these identity signatures, we find that the cell identity signatures, many of them, can be found in the adult brain of the person who donated fibroblasts to make the IPS lines. The signatures are stable. And of course, if we have the adult brain, then you might imagine we have really detailed RNA-seq on the adult brain. And the Allen brain people published 18 months ago a paper saying that you could infer the numbers of neurons of different types being different in different individuals from the sequencing, from the RNA-seq of those individuals. So if you had the IPS lines, then you could look at data of this type and you could correlate the expression of the patterns that you see in the patterning signatures with the presence of different numbers of neurons of different kinds. There might be easier ways of doing this, but it seems to me that in general, in our field, that's a really important question to ask. Because the implication is that from work, let's say we were talking about Parkinson's a little while ago, it's clear that different people have different numbers of dopamine neurons to begin with, and that Parkinson's may be easily, I don't want to trivialize it, but maybe importantly influenced by the number of dopamine neurons you start off with.
For sure, for sure.
Yeah, so this would be extremely interesting to me. Yeah.
There's no question about that, Ron. People say that the decrease in dopamine neurons starts probably in adolescence. It's a very slow process.
Right.
So.
Yeah, you're 75% down by the time you get to the clinic with a shaking hand, right?
So let's go back to dopamine neuron development then. We were discussing it a minute ago in the context of H9 and other cell lines. If we could show for different individuals who have a spectrum of Parkinsonian clinical features, that their iPS cells more or less efficiently make dopamine neurons in the first place, I think you'd really transform our understanding of how to approach the disease, really.
Mm-hmm.
That's terrific. I mean, people should, it would cost some money to do that. It would require some attention. But it seems to me that one function of getting a group like we have today together is to put questions like that on the table and really think about how we might move towards such an outcome. After all, people have large panels of cell lines, and they have genomic sequence, and they have RNA-seq in many cases now from the brains of such people. Anyway, cheers.
We're an hour and a half in, so maybe I'll just show one or two other things and let us get on our way. Firstly, I'd like to show that the result we just had from Nicola and Alex's paper is also live on NEMO, where we have the 9 patterning centers, and then the Jourdan data in the UMAP, the Jourdan data separated by time and cell type. So you can see this HEM signature arising in medial cortical plate fates early on and then falling away as time goes by. And then with these different views, you can actually see it in family by family, even split by cell type. And this shows a little bit of what we just saw, that these differences across the paired father-son pairs is often very different.
So you should sort these lines how we did, right?
Right, that's right. If
because,
we could order, there'd
right,
be 5 going one way,
and
4 going another way. That's right.
I don't know. I mean, I don't know if this can be more informative than what we show, but can be actually something. And overall, of course, we are not correlating this kind of differences with a disease per se. We don't know if this is related to the disease, right? I mean, this is something that we made clear in the paper. We don't know if this actually is related to the cause the disease, right?
Yeah, at this point, it looks like sort of we were saying earlier that if you're at a peak, there's many different ways to fall off the peak, but it doesn't look like autism is always falling off in the same direction. But yeah, how that relates to clinical phenotypes is definitely open.
What's beautiful here is that you can look at different resolution. Like with these visuals, you can look at both like by pairs, by cell types, or like overall across many individuals. And so explore any questions like different resolution all at once.
I think that this has to be also divided by time, because it's rather clear from the differentiation time 0, 30, and 60, right? So I think that this basically confuse a little bit, actually flatten a little bit
Yeah, it'll wash
because
out
what we show in Mato Blanco et al, Alex, is
Right.
just a radial glia at TD0, you know, early.
Yeah.
There's no point to put, in my opinion, based on the data, you know, there is no point to talk about pattern center
At day 60.
centers differentiation day 60.
Yes, I totally agree.
Right, but you're not even 30, by the way. But anyway.
Yeah.
But, Carlo, I just want to, you know, I don't want to. I want to just clarify something before closing the section. I mean, to Ron and Flora, can I share?
Sure.
Be patient, sorry, because otherwise. Ron, you talk about you mentioned about. I just want to say why we consider the ganglionic eminence cells progenitor and case 2.1 positive as pattern centers. We based this on this paper from a fish shell. We're basically showing I mean, it's a review, but basically illustrated it in the mouse. There is basically sonic hedgehog released by the medial ganglionic eminence at very early time point, 9.5. We are a little bit more advanced with the monkey. E37 is around E10, but I don't think we have this kind of early domain, let's say early region. But then sonic hedgehog is basically localized in the medial ganglionic eminence. Okay, so now if you look here, NKS 2.1 is the same. So you see NKS 2.1 in the MGE, in the medial ganglionic eminence here, and there was here. The ventral telencephalon, the homodomain flora, I think that this can be, that's why, you know, NKS 2.1 is before sonic hedgehog or vice versa. I think it depends on the time, but indeed, in the ventral telencephalon, the homodomain transcription factor in case 2.1 is required for the expression of sonic hedgehog in the early MGE through a process analogous to the homogenetic induction of sonic hedgehog in the spinal cord mediated by FOXA2, etc. So that's why, having identified NKS 2.1 progenitors in the ganglionic eminence, because I mean we could not distinguish medial lateral ganglionic eminence, of course, in the monkeys. So in the ganglionic eminence section, we basically identified NKS 2.1 cells, and we called them ganglionic eminence ROSA pattern center.
They're not postmitotic.
They are not postmitotic, no.
Okay. So that's all I want.
No. The NKS 2.1 are not postmitotic.
Yeah.
The neurons, but then we found sonic hedgehog. By the way, in our data, in case 2.1 progenitors, in the stem cells, we could not find sonic hedgehog as we saw in the plot, right? We saw sonic hedgehog in the interneurons. And that's why we said, okay, so this can be considered as a pattern center, and of course interneurons are postmitotic; they're not progenitors anymore. I'm talking about basically this data. What is it?
Maybe it's a characteristic of the human brain. I don't know whether we have sonic hedgehog in mature postmitotic neurons, but we can.
I totally agree. In fact, we see, sonic hedgehog is not in these cells. These are what we consider ganglion. These two clusters are basically what we consider patterning centers. Okay, I mean, in figure 3, we try to understand what is the interaction of these patterning center ganglion ventral cells with the dorsal neural stem cells. And then we found sonic hedgehog in the interneurons. So means later.
Yeah, it could be a later effect. I mean, you have, some people think sonic hedgehog is present in the cerebral cortex too. And some people think NKX 2.1 is present in the cortex too.
I'm
sure anything is possible, but probably at different times, right? Yeah.
So it is basically what we see also in vitro. So basically we see, this is the cell report paper, Carlo, NKX 2.1 is basically, it starts to appear at at DIB17 in our 6 lines. But sonic hedgehog, sorry, don't get dizzy. Sorry for this scrolling. Sonic hedgehog, it's later, you see, it's DIB30.
Yeah.
So in vitro and in vivo, with primate cells, we found the NKS 2.1 is earlier than sonic hedgehog, and sonic hedgehog is expressed by these interneurons. It's exactly what we see in the monkey.
And before both of those, there's a whole packet of organizer genes that show up at day 8, right?
Yeah, right, of course. I mean, these are basically the our ventral lines, right? So these are our ventral lines, yeah.
Yeah, but there is probably also true that you have sonic hedgehog in the notochord in humans much before that.
Much before, yeah, right.
So these are basically ripples, tiny ripples of what has happened in a massive scale.
Yeah.
Quite a few weeks earlier.
Absolutely,
okay?
yeah.
And so you're looking at, it's not because you have sonic hedgehog, you're looking at a single biological effect. Sonic hedgehog may have a number of them.
Absolutely, I totally agree. This is what I said before. I think that, it's basically, it depends on the timing. In our timing, which is slightly after 9.5
so
cells
think
in the
about
mouth.
this. Think about a tsunami wave, okay, which happens early but affects everything. And think about tiny little ripples in the Mediterranean Sea that don't disturb anyone. Okay, that's what we're talking about here. And of course the little ripples are more localized, and the tsunami wave is much larger. I think
One way of thinking about it, Flora, is there's interesting work coming out of Hinnik Victorly's lab at the moment, where he's explaining why you have more motor neurons in humans than in mouse, and where basically it's just a variation of the signaling pathways that are found in the mouse, but in a slightly different group of related cells in the human that maintain them for longer in the proliferative state, which is just kind of what we're discussing. I think I find this really fascinating, this discussion. Yeah. Anyway,
unfortunately, I have to go, so I'm going to have to say goodbye to everyone and thank you for a very interesting discussion.
Grazie a te, Flora.
Thank you.
At the next.
Bye,
Great.
Flora.
Bye.
Great, guys. I think we're closing down on 2 hours in. So thanks, Ron, for opening us, and thanks everybody else for joining and really participating today. Next month, I think Gaurav is going to share some of his Mr. Morphogen exposure experiments with us. So we'll be in this same space. And I think we're going to try to bring a few other things in too to keep our perspective wide on what's going on here. So thanks a lot, everybody.
Before we all sign off,
sure.
Maybe I could just ask everyone, when it seems to me that we want to really keep our working memory active on these topics, and I always find myself thinking that at the frequency we have of once a month, there's a sort of half-life of ideas that declines. So this group, I think, if we're going to really explore some of these topics powerfully together, I would be very open anyway. I'm sure Carlo is the same. To if you have thoughts or ideas that are stimulating, or you want to pursue some of these topics in the NEMO website data, please interact, because I don't see otherwise how we get this done. Now I don't mean we locally, I mean human beings. These topics we discuss require our attention, and attention to things that are not, as I joked about earlier, not already in some AI algorithm. So it requires us personally to decide that it's worth paying attention to a specific idea. Anyway, thanks very much for your attention today. I've really enjoyed it. It would be great to keep it going.
Thanks all.
Thank you. Bye.
Next time.
Ciao,
Thank
ciao.
you. Bye.